This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 195, recorded on February 28th, 2019. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
Where it's 74 degrees. Is that what you told me?
23 degrees Celsius.
It's ridiculous. It's 2 degrees Celsius here. How many miles south of us are you? Like 600?
Probably 800 or so. It's 800 miles to Chicago, so wow. I always equate New York and Chicago at about the same latitude.
We can talk about the weather because Elio and Michelle are not here.
Yeah.
They don't like it.
They don't like talking about the weather, but it seems from our letters that they do like hearing about the weather, especially Elio's weather report of San Diego when you're in England where it's wet and dank and cold.
You bet.
That was one of the letters today, I think. They gave us their weather report.
If you haven't done so already, please take the TWiM poll. We want to know a little bit about you, our listeners. It's very short, 12 questions, takes 5 minutes to answer. Nothing hard, nothing about microbes at all. And this survey helps us collect data for getting sponsorship, which we'd like to do to try and grow the podcast. You can find it at asm.org/TWIMPoll, asm.org, TWIMPOLL, all one word. And we thank you for that, and we thank you for listening. Today we have, of course, papers for you, stories, 2 stories, which are really cool. And after that, we will give away a copy of Bugs as Drugs. We had a lot of people write in. 41 people wrote in. And, you know, Michael, I do this from time to time just to hear about our listeners.
Yeah.
Because otherwise we don't hear too much. So stay tuned for that.
Only when we're wrong.
Only when we are wrong. That's fine. It's good to correct us for sure. Absolutely.
Yes, especially me.
All right. So I will start off with a snippet, which is quite interesting. It is a letter in Nature Medicine. It's called Identification of Pre-Existing Adaptive Immunity to Cas9 Proteins in Humans. And this comes from the Department of Pediatrics at Stanford University and a company called Integrated DNA Technologies in Coralville, Iowa.
Before you get into the paper, I just want to mention that this morning I'm one of the representatives to our faculty senate. And I'm at an academic health sciences center. And so one of the discussions elements that came up this morning is that we need to be able to explain the CRISPR-Cas to our patients that are coming in and asking questions. And this was from one of the individuals in the College of Nursing who brought this up. So this is actually making it into mainstream medicine where the general public are curious about how this is working. So I think it's timely that we're talking about this paper because there are many companies out there developing medicines that the general public is learning about through the popular press.
Mm-hmm. Well, this is going to be used in people one day. So
yes,
it's important. And it is CRISPR Cas9, and it is a repurposed defense system from bacteria, archaea, that is huge because it allows you to modify DNA sequences specifically. And so the CRISPR part is a short sequence that you can use to target any gene. And then Cas9 is an endonuclease that will recognize the target by virtue of the duplex, and it will cut it.
Magic scissors.
Will cut it. There are many of these CRISPR-associated sequences, Cas, which are these endonucleases, but a few have come into be used the most. And these have been slightly modified by a number of people to be easier to use. But in fact, it's really easy. Let's say I have a cell line and I want to disrupt a gene, I make an oligonucleotide of a certain length. That'll be the guide RNA, a short guide RNA. And I put it into cells along with a plasmid that makes Cas9. And the guide RNA will attach to the target, and the Cas9 will cleave it. And then the cell will go, oh, there's a break. Let's fix it. And then usually makes a mistake in fixing it. And there you have a mutation. Now, if you wanted to replace a gene, say you wanted to replace a defective gene, you could put in a good copy, and often the cell will use the good copy to replace the old one when it's trying to replace it, when it's trying to fix it. And so it's really remarkable the things you can do. And a couple of the applications include taking stem cells out of patients and either removing a deleterious gene or putting in one that's not there using CRISPR Cas9 systems. And then you could put those cells back into the patient and they would re-establish themselves in various tissues. Or you could inject a vector, a virus vector, into the patients that delivers CRISPR Cas9, and it will go and fix whatever it is that you need to fix. So these are just entering clinical trials, and they're going to end up in people at some point. And so this,
one of the examples that I think they used was talking about addressing the issue with sickle cell disease, where the individual is not able to make the proper hemoglobin, and they're using the CRISPR system to effectively restore the defect, so that the individual will indeed make the right form of hemoglobin, so that they will no longer suffer from sickle cell anemia.
Right. So this has the potential to correct a lot of genetic defects and make humans live even longer and get other diseases that weren't planned during evolution. No, I digress. We live a long time, and that's why we have other issues, but that's another story. So the commonly used Cas9, so Cas9 is an enzyme, all right, and it is either from Staph aureus or Streptococcus pyogenes. And those are the two bacteria that from which most of the Cas9 proteins are used. And they have different advantages. For example, one is smaller than the other. The Staph aureus is smaller, so it's used in applications where the gene has to be put into a virus vector because you don't have a lot of room in these virus vectors. And the S. pyogenes Cas9 has also been shown to work well as long as you don't have a size limitation.
And they use that, they mentioned, for preclinical editing.
That's right. Now, these are two common commensals that can also cause disease. 40% of humans are carrying Staph aureus, and 20% of school-age children have Strep pyogenes.
The beast that causes strep throat.
Strep throat. So it's rather common. And so the question here is, well, since these bacteria are so common in the human population, is it possible, since Cas9 comes from these bacteria, is it possible that we have immunity to Cas9? And that's an important question, because if we do, it could impact the efficacy of the CRISPR Cas9 modification if it were ever used on a person who has immunity. And we know in gene therapy trials, when we use virus vectors, pre-existing immunity is a real problem. You know, if you have neutralizing antibodies to the vector that you're given, it's going to limit the efficacy. Or if you have cytotoxic T lymphocytes, that are specific for the transgene, they will go and home on cells that produce the transgene and lyse them. And so this is known to be a problem in other clinical trials of gene therapy. And so it could be that if you had cytotoxic T cells against Cas9, it would destroy the cells that have the Cas9 in them. And those would be ones that are presumably having some therapeutic value to them. So that could be a problem. So they did a study here where they looked for antibodies and T cells specific for Cas9. They did a couple of different things. The first study, they looked for antibodies against the two Cases, the strep and the staph Cas. and they get serum from human cord blood. And that's, of course, from the cord that is connecting the mother and the baby. And that reflects the mother's IgG, the cord blood. And then they simply did Western blots to ask, they run proteins on Cas9 proteins on a gel, transfer them to a membrane, and probe the membrane with the antibody, with the serum. And they see that 86% of the samples were positive for the staph Cas9, and 73% for the strep Cas9. So that's pretty high. And then they looked at serum from peripheral blood next. That was from, this first study I told you, they looked at 22 cord blood donors. Then they took serum, peripheral blood from 12 healthy non-pregnant adults, and they found staph antibodies in 67% of donors and strep and Cas9 antibodies in 42%. So kind of mirroring the prevalence of the bacteria in humans, right?
Yes.
And then they made an ELISA assay to say they fix Cas9 protein on a plastic, and then they pass the serum over it, and then they detect any bound antibody using a second antibody. And they found in 125 blood donors, they found antibodies against both Cas9s in 125. I don't know what the percentage is there. Say it.
It's when probing against each antigenic protein, the Staph aureus was 1.34, the Streptococcus was 1.092, and tetanus toxoid, for which most of the United States is vaccinated, it's 2.0. So that gives you an idea that the antibody is literally, we have antibodies at half the rate that we have antibodies to tetanus toxoid.
99% of donors had tetanus toxoid antibodies.
And 50% didn't.
Yeah.
50% had antibodies against one of the both of the Cas products from Staph aureus and Streptococci.
78% had Staph Cas9 antibodies, and 58% had the Cas9 from Streptococcus.
Yeah.
All right. So then they said, how about T cells? How do you look for T cells? Well, you take peripheral blood mononuclear cells, and then you add Cas9 protein to them, and a certain number will take them up, and they will react in different ways depending on whether they're specific for it or not. So they can make cytokines, and they can display certain molecules on the cell surface. And so...
and then you can use flow to detect them.
You could use flow, and there are a variety of assays. ELISpot will detect interferon gamma, which is produced by T cells when they react to an antigen that they see, and that means that they're specific for it. So in the ELISpot assay, 78%, 55%, and 33% of donors were positive for T cell reactivity to tetanus toxoid. That was the highest. Staph Cas9 and Strep Cas9. So there are T cells in the population specific for this. Another assay, a cytokine response, 50%, 66%, 44% for tetanus, staph, and strep Cas9. And then looking at these surface activation markers. So in other words, if a T cell is present that is specific for Cas9, it will become activated and display certain markers on its surface, which you can detect by flow cytometry. And by that assay, the percentages are much lower, less than 1% for all. They took some of these cells that seem to react, T cells that seem to be specific for Cas9, and they purified them by flow cytometry and re-challenged them with Cas9, and they again responded, showing that they are certainly specific for the antigen. So basically, they did 4 assays, ELISpot, intracellular staining, and then these 2 surface molecules. And if they decided if they would say a person is positive, if it were positive in 3 out of these 4 assay. So using that threshold, 100% of donors are positive for T cells against tetanus toxoid, 78% against staph Cas9, and 67% against strep Cas9. So there's clearly...
It doesn't bode well for the adeno-associated Cas9 system.
It's clearly immunity, pre-existing immunity in the human population. And, this is, they mentioned that 2 other groups have also reported on this. This is something that had to be done. But, the clinical trials are still plowing full speed ahead because of the promise of this. But this is an interesting issue. So if you have, depending on how you're going to treat someone, this could be an issue. And they say it may not be a problem for ex vivo therapies where you take cells out of the person, typically stem cells, and then you modify them and then put them back in. So that may not be a problem. But because they say that any peptide fragments of any protein delivered ex vivo are typically only expressed transiently, and they may not be, the Cas9 peptides may not be on the cell surface when they put them back in the people, especially if it's delivered as a short-lived complex. So that ex vivo, not a problem. More likely is, as Michael said, if you want to infect people with an adeno-associated virus vector that's going to deliver CRISPR Cas9 to certain places, then those cells, then there may be memory responses in those hosts, and they may destroy any cells that have Cas9 on the surface. Antibodies, I can imagine antibodies reacting with proteins as well on the cell surface or soluble protein and causing problems. And they say maybe you get organ toxicity. So I suspect that what they will do is look for that in the clinical trials, right? Instead of trying to intercept ahead of time, they will probably just say, let's look and see if there's any toxicity. But they say here some solutions include maybe we could give these individuals steroids, just like we do when they get transplants, right?
Or even blood. When you receive a pint of blood, if you need it, they often give you something to suppress your immune response. They typically give you Benadryl if you're hanging a unit of blood so that you don't react badly to the blood, even though it's already typed and crossed to match you as closely as possible. They suppress your immune system with some Benadryl and some other agents to make certain that you don't react badly to this foreign material being introduced to you.
And they say you could deplete these Cas9 positive immune cells. That'd be a lot of work, I think.
Well, you just pass them through a flow machine and it would sort them, and they would be Cas negative cells that you would get. But as you point out, if any of the peptide fragments then surface on the cell subsequent to sorting, they could be reactive. And, strep throat is one of the classic diseases that it has a rheumatic variant that we know as rheumatic heart disease, where because it cross-reacts with one of our antigens, principally on our heart tissue, we can develop rheumatic symptoms, which means there's no infection. Your immune system's just been awakened to the invader, and then it goes out and it attacks anything that sort of looks like it. And, it's a very slippery slope. Anyone who has a rheumatic complication well knows the slippery slope of a subsequent strep infection.
Their other suggestion is to say maybe we should look for or use Cas9 orthologs from bacteria that don't infect us. And I'll bet people are looking for those or already have them and are working on it. That would make a lot of sense, right? Instead of having to worry about having issues here with the immune responses.
But again, it depends on how similar the Cas proteins are to each other antigenically. And short of not knowing how they are similar or dissimilar, we don't know the answer to that because, you can have different species, but it cross-reacts. Again, that's why streptococci are so evil to us, is because our heart antigens look just like a strep does.
So maybe they could take
molecular memory.
Maybe they could take the strep or the staph Cas9 and ablate all the major antigenic sites and hope it doesn't mess up the protein.
Yeah. we certainly know how to engineer proteins better than we did 20 years ago when the tragic case of Jesse Gelsinger happened.
Yes. Jesse was receiving viral gene therapy and died of a massive
immune
reaction, essentially, which destroyed his organs and killed him. They gave him too much virus, most likely.
Yes.
And that was at UPenn. And the whole field suffered as a consequence, as it should, because they had to stop and back off. But, clinical trials, they like to be gung-ho, don't they?
They do indeed.
Well, you have to cure diseases.
People are anxious for a solution. And, the go slow just simply adds to the cost, but it also lowers the risk. And it's an issue of what your risk tolerance will be.
Right.
And if you have a low risk tolerance, you want it to be 100% safe.
So their last sentence is important. We believe our findings will stimulate crucial discussions in the genome editing community regarding safety and efficacy as this technology is translated to humans. So everyone doing these studies, please read these papers and act accordingly. All right.
So that is a fantastic snippet. Thank you, Vincent.
It's a perfect snippet, isn't it?
Yes.
And it's important as well. So there you go. And unfortunately, it's behind a paywall. It's in Nature Medicine.
It's not open access. They didn't make this one open access. Let's see. No, it's not. It doesn't have an open access symbol on it.
No, it costs a lot of money to make your paper.
Yes.
Open access.
I know.
Did you make yours?
Especially in Nature.
Yeah. All right, Michael, you have an interesting one.
I do indeed. And this one made headline news in late January of 2019, if you're behind on your TWiM catalog. And the title of the paper that we're going to work through this afternoon is Porphyromonas gingivalis in Alzheimer's disease brains. Evidence for disease causation and treatment. You heard it right. Treatment with small molecule inhibitors. And this is from a very large group of 26 authors. The first two authors, Domini and Lynch, are co-first authors, and they're from a company in South San Francisco called Cortex-Zyme. And there's a group of others from various other institutions, and I commend you. It's truly a global project. There's folks from Poland, New Zealand, Australia, Kentucky. It's truly an international paper. It was published in Science Advances on the 23rd of January. And in brief, a microbe that has been implicated in chronic periodontitis, or gum disease, if you will, appears to be a player in Alzheimer's pathology. Say what? The flossing may prevent Alzheimer's? I
thought that would stimulate it.
There was a big stink a few years ago in the dental community that flossing showed no benefit one way or the other. And now here is a periodontal pathogen. It's really quite remarkable. Now, for many years, there have been hints that microbes may have been are implicated in the development and progression of this life-crushing disease. However, evidence for causation, or if you will, the fulfillment of Koch's postulates for direct microbial involvement for Alzheimer's disease has not been great. But today, we're going to be offered an opportunity to explore new data that actually implicates this oral microbe, which happens to be a black pigmented, asaccharolytic, non-motile, gram-negative microbe that requires anaerobic conditions for growth, which implies a low electromotive force and the presence of heme and vitamin K in its nutrient list of things you must feed it.
Do we all have this in our mouth?
Principally after about the age of 35, or if you have poor oral hygiene, it begins to lay itself down in dental plaque. And what you have to know about Porphyromonas gingivalis is it's got special nutrient requirements due to the fact that it's asaccharolytic, what that means is it subsists on small peptides cleaved from host proteins. So this is the trick of this microbe. It eats us. And what this tells us is that this microbe has this panoply of virulence factors. It's got proteases. It's got at least 12 virulence factors that I know of. And it thrives as a component of a polymicrobial community where other players are doing things in its local environment. And it's principally a passenger of dental plaque, which is why we go to the dentist in developed countries every 6 months to effectively scrape the biofilm that's associated with our teeth off to prevent these microbes. And gingivalis is really a pretty mean actor because its virulence factors can effectively subvert our leukocytes. And what they do in our oral microbiome is they foster something referred to as microbial dysbiosis, which simply means things aren't normal. So we need to ask, where is niche one if Porphyromonas gingivalis is truly indeed implicated in Alzheimer's disease? Because you don't have to be a healthcare provider, a physician, a microbiologist to know Alzheimer's disease is a disease that affects principally the brain. But anyone who has, over the age of 35, who has visited their dentist has had the god-awful perio exam where they take that really sharp stick and they poke it up your gums. And as we get older, our gums recede, sort of like my hairline. And the tone of our gums gets a little flabby, like my imaginary 8-pack abs. So Porphyromonas is one of the red oral microbes, indicating that it's involved in the pathogenesis of periodontitis. Now, the perio guys, they classify the organisms that are responsible for disease in the oral cavity by color. So the red ones, as you can well imagine, are really bad actors. So Porphyromonas is in the red box of the Sokransky catalog of what's actually going on. So what this very diverse group of investigators will show in this paper, and they were very careful in how they laid out their story, is they have identified the presence of this keystone pathogen in chronic periodontitis. And that's the operative word here, is Porphyromonas gingivalis is the player that becomes chronic in individuals that have this affliction of their gums. And that this keystone pathogen of periodontitis is in the brains of Alzheimer's patients. So we're not talking mice today, we're talking people. And of its virulence factors, Porphyromonas gingivalis has this toxic protease named a Gingipain. And this Gingipain was also identified in the brains of individuals suffering from Alzheimer's. And this is the remarkable bit. And the concentration or its levels were correlated with the concentration or the presence of tau. And I didn't know much about neurobiology until I began to dig into this paper. And tau is, of course, implicated in Alzheimer's disease because of how it forms plaques and tangles, as well as ubiquitin pathology. And so ubiquitin, we know, is to identify bad actors and to get rid of them. And so this Gingipan or virulence factor, which is a cysteine protease, and remember, this microbe needs to destroy our proteins in order to live. This is effectively the story that's going to evolve. Now, they take us back into animals for a short while, and they tell us in animal studies conducted in mice, this oral infection by this pigmented microbe results in brain colonization in mice and an increased production of human amyloid beta peptide, which is, of course, a hallmark of Alzheimer's disease, because upon its deposition, it leads to this formation of these amyloid plaques and the pathology that we so tragically associated with Alzheimer's. The third piece of their story is that this toxic protease, or the Gingipan, was found to be neurotoxic in vivo and in vitro, resulting in the detrimental effects of tau. So before reading this paper, I hadn't grasped quite the significance of tau. I knew about plaques, I knew about tangles, but not the genesis of the disease. So here's where I learned that by virtue of the fact that these gingipans attack tau, which is a protein needed for normal neuronal function, I appreciated the significance of this microbe leaving the niche of our oral cavity, even if we have periodontal disease, and crossing into our brain where the microbe has starts an infection. The protease effectively begins to literally consume the protein in our brain, which is then responsible for the development of the Alzheimer's disease. Now, in their attempt to address causation, where they can literally show that if you knock out the Gingipan activity, they developed small molecule inhibitors. And they specifically targeted two of the gingipans. Porphyromonas gingivalis has two flavors of gingipans. One is a lysine variant, and the other is an arginine variant. And they learned that through gingipan inhibition, they were able to reduce first bacterial load of an established Porphyromonas infection. Now we're back into mice, with the inhibitor being able to block production of the amyloid beta peptide, which is this necessary component for plaque formation, which in turn, adding the inhibitor of the Gingipan, then reduced neural inflammation. And we know that Alzheimer's disease also has a component of our immune system coming in, and neural inflammation. Any inflammation we appreciate now is probably bad and not conducive to extending our lifespan or our quality of life. And perhaps most remarkable, they were able to demonstrate the rescue of neurons in the hippocampus, where Alzheimer's is thought to begin, because it's involved in the hippocampus is involved in the consolidation of information from short-term memory to long-term memory. And it's also involved in spatial memory, which enables us to navigate, which is oftentimes why one of the first symptoms associated with Alzheimer's disease is you forgot where you put your keys. You know, the ability to navigate and find your keys that you have somehow misplaced. So collectively, the paper makes the argument that the Gingipan inhibitors could be valuable for treating this Porphyromonas gingivalis brain colonization. And if you're thinking like a microbiologist, then what this group has simply done is created a new antimicrobial, albeit a very specific and targeted antimicrobial against two proteins that this microbe makes. And they're anticipating that it will be able to treat the spread of this disease and hopefully be able to reduce suffering for both the victim of the Alzheimer's disease and the families caring for the sufferers of this infection. And this will then be a true miraculous and magic bullet. And the way they end the paper is that the magic bullet has extremely high bioavailability where the infection starts in the oral cavity. And secondly, its bioavailability is also very high in the central nervous system. So then it does the double duty of both retarding the growth of the actor that starts the event, first in the oral cavity, and also limits the factor responsible for the generation of the pathology And so now we have a different class of antimicrobial, and potentially we can limit the neurodegradation seen in Alzheimer's patients. And so it's really a remarkable paper that they go to great lengths to walk us through. And there's 10 sections to the paper that are really laid out quite nicely, establishing the causation, or if you will, fulfilling Koch's postulates that Porphyromonas gingivalis is indeed involved in Alzheimer's disease. So they first start us off by showing us that Alzheimer's disease diagnosis correlates with Gingipan load in the brain. And principally, they did this experiment the way you might imagine this experiment would be done. They took sex and age-matched brain tissue cores from sufferers of Alzheimer's disease and also normal individuals. So these individuals have passed away. They are dead. And then using Gingipan-specific antibodies that were targeting the two flavors of Gingipans, the lysine variant, which they refer to as K, the single amino acid abbreviation for lysine, and then R for arginine. So it's RGP and KGP. And they determined that the Gingipan load was significantly higher in tissues from Alzheimer's disease brains than non-demented brains. They then next looked for tau, and they found a significant correlation between the arginine Gingipan and tau load. And this is important, again, because tau pathology has been shown to strongly correlate with cognitive impairment in Alzheimer's disease. They then go to the next level, take us to tissue microarrays, where they stain for ubiquitin, which is, again, a small protein tag that marks damaged proteins for degradation by proteasomes. And we're able to see it accumulates in both tau tangles and Alzheimer's disease plaques, learning, again, a significant correlation between the Gingipans and the Alzheimer tissue microarrays. And so these findings were then consistent with the concept that Alzheimer's disease and the Gingipans are strongly linked. They then go the next level, and they show the correlation analysis between Gingipan load and tau load, and between Gingipan and ubiquitin in non-demented control samples. And here's where they learned a continuum of Gingipan and Alzheimer's disease pathology already present in controls. So what this is telling us is that this infection is probably chronic in our brains. This was my uh-oh moment as I was reading this, going, I got to go to my dentist and make certain that, I got good periodontal health, because
what fraction of the controls again?
Let's see, where is that? That's in figure 1G. I don't know off the top of my head.
1G normalized to control.
It's blue and red. So it looks like the blue is a little bit, it's on a logarithmic scale, normalized to control. And it looks like the reds are moving up. So it looks like it's an order of magnitude difference. But it is indeed going up.
So according to the paper, only about 25% of healthy people have P. gingivalis. So
right.
Maybe you should have a serum antibody test and see if you're infected.
Well, they take us there. They do indeed. That's part of their story as well. They have a PCR, and I'll jump to that discussion. They have a PCR assay that specifically looks for the presence of Porphyromonas gingivalis using 16S ribosomal RNA gene, as well as another gene, HMUY. And HMUY and HMU-R liberate heme from host heme binding proteins through the actions of proteases. So if they detect the genes for first the 16S ribosomal RNA gene for PG, as well as this heme protein, which is a defined growth requirement for the microbe, they say that they can detect it in both saliva as well as in CSF from cerebral spinal fluid.
Mm-hmm.
So that's effectively a very important point, because as you begin to think about a clinical trial, the fact that you have a very sensitive real-time PCR assay for measuring concentration of, if you will, the microbe present in the CSF, you can gauge whether or not the small molecule inhibitor that they're going to take us to is actually working and reducing, if you will, the infectious load. So you have a way of detecting whether or not something is working.
Of course, you can do MRI to measure the Alzheimer's progression too, right? So you could take people. I would guess the clinical trial is going to be that take people with early onset Alzheimer's, beginning, not too advanced. They'll do a scan, a brain scan, to establish the baseline. Then they'll treat them, and a year later they'll do another scan. They'll measure all the measurements that you do for Alzheimer's and see if there's any improvement.
That is indeed the case. And in fact, that clinical trial is presently underway, and the closing line of their paper is to clinicaltrials.gov.
Oh, cool.
So you can actually go and read what their clinical trial is all about. And so that's the closing line of the paper, and we can probably put that in the show notes. I've lost the clinicaltrial.gov in the paper, but we can put that in the show notes certainly for you. And this was really a beautiful paper because it's a mixture of they're testing a hypothesis that this microbe is implicated in the disease. They develop data that equivocally show that there is an infection, and the Gingipans are implicated in the development of the pathology associated with the phenotype of the disease as we presently understand it. And their PCR test that they're looking for PG, it works at a sensitivity level of between 10 and 50 copies per microliter of cerebral spinal fluid. So it's a pretty sensitive assay for looking for the presence of P. gingivalis DNA in the cerebral spinal fluid of clinically scored and identified Alzheimer's disease patients. And any student of microbiology knows and appreciates that cerebral spinal fluid is a window into the neurological infections of the brain and the neuronal system. And the fact that they were able to detect it both in CSF and saliva is really pretty cool. The next thing they took great pains and showed us is that tau is actually fragmented by the activity of the Gingipan. And these experiments illustrate how bad Gingipans are, and it really implicates them in our understanding of Alzheimer's disease pathogenesis. Simply put, tau is fragmented by this protease, the Gingipan, and this truncation of tau has truncation and fragmentation have been proposed to play this key role in the formation of these insoluble and hyperphosphorylated forms of tau in the Alzheimer's disease patients. And again, it was simply following exposure to an extremely low concentration of the Gingipans, namely 1 nanomolar Gingipans, that they were able to see tau. So then they go after making the inhibitors to ask the question, are these small molecule inhibitors neuroprotective? So now we're going back to cell culture. So here the experiment is, are the neurons in the form of cell culture, when they're exposed to the Gingipan, to both flavors of the Arg or Lice Gingipan variant, and what they saw is, as you might guess, is as measured by aggregation. But when they treated the proteases with an inhibitor, and they started out with iodoacetamide, which is an irreversible cysteine protease inhibitor. Principally, it knocks out cysteine proteases by preventing them from forming disulfide bonds. And they then developed a library of potent and selective, either reversible or irreversible small molecule Gingipan inhibitors. And they settled on two that have an extremely low median inhibitory concentration of less than 50 picomolars. So the pharmacologists are really excited about concentrations that act at an extremely low level. And they did the microbiology for me to verify that the new inhibitors work. They took the cell line, they infected it with Porphyromonas gingivalis at a multiplicity of infection of 400 bacteria for every one neuronal cell in culture. They let it cook for 48 hours. That in turn killed half the cells. But when you add either of their irreversibly binding inhibitors, either Core 286, which was the arginine inhibitor of the Arg Gingipan, or Core 271, they were both effective in blocking Porphyromonas gingivalis from killing neuronal cells in culture. What I found was the most remarkable piece of this experiment, though, is they had two antibiotics. The first was a gyrase inhibitor, moxifloxacin. So that's going to address DNA synthesis in the bacterium. And then a protein synthetic inhibitor, doxycycline. And even at this MOI that routinely reduced bacterial survival in vitro, those two antibiotics failed to protect their neuronal cell line. So it either tells you that this organism, this Porphyromonas gingivalis, is throwing these gingipans out like crazy, and they're responsible for the killing. And that's, again, their next experiment, where they took the gingipans and they were able to demonstrate they were neurotoxic. Now we go from cell lines into mice, and they ask the question whether the inhibitors can penetrate the brain and prevent gingipan neurotoxicity. So here's where they give the poor mice an infection. Here, 8-week-old BALB/c mice given a single administration of gingipan inhibitors via a combination of the two. They inject them subcutaneously. Once the drugs are on board, they then inject the hippocampus of the animals with either a combination of the two gingipans, and that they learned that the mice that were injected sub-Q had a significant number of gingipan-mediated degradation neurons. But if you had the inhibitors on board, it was able to block the neurodegeneration.
So they're injecting the mice with bacteria, not gingipan.
No, gingipans. They inject the inhibitors sub-Q, and then they stereotactically inject the gingipan into the brain, the hippocampus region of mice, to effectively then sack the mouse, ask the question, did it destroy the gingipan? The gingipans destroy the hippocampus. So if the inhibitor is present, it didn't destroy. If the inhibitor, they're in the control situation, it did. They then did the next experiment with oral infection of the mice with P. gingivalis. This first then resulted in a brain infection, induction of the amyloid beta protein system. And again, these were in BALB/c mice. And again, they're able to show that these inhibitors were able to do the thing they needed to do. So they do more brain chemistry for us. And as they begin to walk us through this, they investigated whether this amyloid beta 142 protein or peptide was able to interact with P. gingivalis and decrease its viability in vitro. So they're asking the question, is the human response of making this amyloid beta protein, it's the human's attempt to try to inhibit the infection in the brain. And so here they incubated the amyloid beta protein with gingivalis, and they learned that the amyloid beta protein co-localized with the gingipan on the surface of the bacterium. And because amyloid beta protein is known to disrupt cellular membranes, they hypothesized that this amyloid beta protein might disrupt the integrity of P. gingivalis to cause the cell death. And as you can probably guess, they found the proportion of dead and dying Porphyromonas significantly increased after incubation with this amyloid beta protein when compared to a scrambled version of this peptide or phosphate buffered saline. So now we see why the human is actually making, if you will, this peptide, because it's responding to an infection.
Yeah, there's also viral results that suggest that as well. For herpes simplex viruses in culture and in mice, you can show that the amyloid is actually an antiviral response, and here it's an antibacterial response as well.
Mm-hmm.
So it's killing us.
Yes. So we're taking out our cells.
Yep.
So then they took oral administration of the lysine inhibitor, the lysine gingipan inhibitor, and they were able to demonstrate that this was effectively able to treat a brain infection and prevent loss of the hippocampal region, the interneurons in vivo. Again, they're using straightforward designs in their experiments. They orally infect the animal by giving them 1 billion colony-forming units of Porphyromonas gingivalis. They give this concentration of bacteria to the mouse every other day for 42 days. Then they let it rest for 28. They then start treating with the inhibitor at day 36, and they give it the inhibitor from day 36 to 70. And they were able to identify the bacterium in the brains of all the infected mice at day 35. They're only beginning to give the inhibitor at day 36. And with the concentration of bacteria increasing in the brain by day 70. So this is the hallmark of a real God-fearing infection in our brain. When they add the lysine gingipan inhibitor, Core 271, that has a high oral bioavailability and this significant CNS penetrance, and this was only administered twice daily orally, they found that this first reduced the bacterial load in the brain compared to the positive control to the levels at baseline at 5 weeks. When the arginine inhibitor was administered sub-Q, they were also able to reduce the bacterial bioload in the brain at 10 weeks, but they learned that it wasn't as effective as the lysine inhibitor that they had developed. And then they do the gold standard microbiology. They give antibiotics. So they give moxifloxacin. So moxi was found to be beneficial in preventing an increase in the concentration of Porphyromonas in the brain between days 35 and 70. But here's the kicker. But it was not effective in reducing the concentration of Porphyromonas below the 5-week baseline, which the lysine inhibitor was found to do. When they mix the two of them together, the Core 271 with the moxifloxacin or Core 286 with moxi, this didn't improve efficiency over the Core 271 by itself. And their final experiment is they make even a better inhibitor, and they call this one Core 388. And this treatment regime showed dose-dependent effects on Porphyromonas gingivalis infection and tumor necrosis factor levels in mice, akin to what you probably see with herpes infection in the brain. And so based on the results from their former experiment, they then developed an experimental paradigm using a defined growth medium and asking what's actually going on in this. And they're looking to answer the proverbial question of every microbiologist. Will the microbe develop resistance to this peptide? And like most antibiotics, this microbe can develop resistance in as little as 12 passages to moxifloxacin. But they did not see the development of resistance to their new, if you will, antimicrobial inhibitor, Core 388, in two separate independent assays over the course of their studies. And, they have an investigational new drug application for Core 388, and the compound is currently in clinical trials. So summing up, there's evidence, pretty good evidence, I think, that Porphyromonas gingivalis plays a role in Alzheimer's disease brains. It's a highly selective antimicrobial that they developed and presented can limit the further development of growth of the microbe in the brain of least mice and prevent the concomitant pathology that develops. At the end of the day, we still need to ask some questions. What's the spectrum of activity of this new antimicrobial? Will it cross-react with other Gingipan-like protease molecules in the oral milieu and change the ecology badly? Will this inhibitor cross-react with anything important in our brains, thereby altering the chemistry or the pathology of Alzheimer's disease? And while they address this in the manuscript, I'll leave you with the sentiment of Michelle Vanderpool, the microbiology and serology supervisor at the Rady Children's Hospital in San Diego, who challenges us all to recall the Borg from Star Trek's Next Generation fame in her signature line of her emails, where she tells us that the Borg were wrong. Resistance is not futile. It's inevitable. So on behalf of the 75 million baby boomers, I'm hoping the Borg were right and that resistance against these small molecule that this group developed will be futile. I hope that Porphyromonas doesn't become resistant to this small molecule, and so we'll have an effective treatment for Alzheimer's disease.
Well, you can always develop other compounds too, right?
That's true.
So this compound, Core 388, has already been in 2 trials for safety at different doses. So it's the real one now is in, which is not yet recruiting, but it's listed in clinicaltrials.gov. I'll put a link in the show notes. It is a study of Core 388 in subjects with Alzheimer's disease. So that's going to be a true, functional study. And so what they're going to do, they're going to enroll 573 subjects with a documented diagnosis of mild to moderate Alzheimer's disease. They will then have a variety of things measured, as we see, and then they're going to be given this drug for a number of weeks. I'm not sure how long. It's going to go for quite a while, and they will see what happens to them. Very interesting.
I tell you, this was a fascinating paper to take apart, which is why I spent so much time taking it apart for the group, for the audience today.
Now, I have a couple of comments. First of all, so their percentages of this P. gingivalis in their Alzheimer's patients is very high. It's over 90%.
Oh, yeah.
Which is remarkable because, as they say, there are other microbes that have been implicated in Alzheimer's. And over on TWiM, we've done a couple of episodes in the last 6 months.
With herpes.
With herpes viruses and implicated in Alzheimer's. And there's a clinical trial ongoing, enrolling at the moment to give AD patients herpes antivirals to see if it does anything. So I suspect that there's going to be multiple microbes involved.
Oh, yeah.
With the etiology of AD. This may be one of them, but there'll be others. There may be other bacteria, other viruses. But these data are very compelling.
Well, they didn't say this is the only microbe implicated. It does indeed fulfill, in my estimation, Koch's postulates that you can, if you inhibit this, if you inhibit the microbe, you will inhibit the development of pathology, at least in mice. But the gold standard is in the mixed environment of a true human experience where you can have exposure to herpes infections and all of the other things. You could well imagine developing a cocktail of core 388 and acyclovir to control herpes infection.
Yeah. The other thing I wanted to bring up is that a genetic risk factor for Alzheimer's is the gene encoding apolipoprotein E4. It's the greatest genetic risk factor for sporadic Alzheimer's that doesn't have that just occurs randomly. It's not,
and that's
in your
for
family.
atherosclerosis.
Yeah, so that's a protein in the blood that carries cholesterol. Now, it's been proposed that Gingipain cleaves ApoE4 and makes neurotoxic fragments, and that's why ApoE4 and not other apolipoproteins like 3 or 2 may be a risk factor. So that's pretty interesting as well. So I think this is compelling, although I do think there are going to be other causes. So, this may not be the one. Figuring out which treatment, right? So you have patients with Alzheimer's. I guess you see, do they have P. gingivalis infection? If so, you treat them. If they don't respond, maybe it's something else, right? I mean, we still have a long way to go, but it's pretty interesting.
Well, the question is, if you have active periodontal disease, you just put somebody on this medication, hoping that it will, one, control the advancement of periodontal disease. Do we have a drug that has high bioavailability to the oral cavity that could actually control and limit periodontal disease? Because gum disease by itself is no picnic, because you ultimately lose your teeth.
So where do we acquire the P. gingivalis? If we don't have it and we acquire it at a certain age, where is it coming from? Another person, right?
It's coming from another person, or it's being introduced with food, or it's just out there.
It's when you put your toothbrush on the hotel sink. That's where you get it.
Next to the toilet. Next to the toilet when we flush, generating aerosols.
Oh, man. Life is very dangerous, isn't it?
Well, we at least now have a better understanding of this, and it gives us a path forward. I know this paper was a little bit long by TWiM standards, but it walks us through, and there was a large group of workers involved in effectively presenting these data to us today.
So we had gotten a letter from Sue Ellen, who wrote, this popped up in my newsfeed yesterday. It's an article in the New Scientist about this paper. Wanted to send it to my TWiM microbe experts for review. I have no idea how well the actual science supports the claim that P. gingivalis causes Alzheimer's, and that drugs that suppress or kill this bacteria can improve cognitive functioning of AD patients. I'm always a skeptic, but I lack the education to be able to dive into the actual data. So I thought I'd pass it along to my panel of experts, or at least a lesson in how to tell the difference between media hype and veritable scientific results. As you guys are always saying, mice lie and monkeys exaggerate. Well, Swell, and I think the data look pretty good, and the key here is going to be the clinical trial. I mean, it's good enough to go into people, I think, and it is. So, but I also, as I said,
I got through the safety trial.
I also think, this drug passed safety test, no toxicity and so forth. So I do think that there are going to be other causes. So this is not going to cure it all, but it might get a good fraction, and then so we'll see. It's very good. All right, let's give away a copy of Bugs as Drugs. We had 41 entries.
Wow.
From people like Cindy, who's a second-year microbiology student, Julie, who teaches microbiology at a community college. Walter did his master's in neuroscience, but switched to microbiology for his PhD. Joshua is in England, and he loves the weather segment. Currently enduring between 5 and 8 Celsius, which is borderline intolerable for us grumpy Brits. Chelsea says, I'm listening to TWiM as I prepare Kirby Bauer plates for the microlab I'm teaching this week. She's at Parkland College. Sam is a chemist who loves, he's here at Columbia Law School, actually.
Oh.
And he's a chemist, but he loves our podcast. Christopher is a PhD student at Indiana University.
Where I got my degree.
Isn't that where Carl Woese used to be?
No, Carl was at the University of Illinois. Norm Pace was at IU.
All right. I get them confused. Now, what you got your undergrad at Indiana U?
No, my PhD.
PhD. So I did a TWiM there a couple of years ago.
I saw.
Remember?
Yes.
TWiM and a TWiV, actually. Yeah. They cupped two of the TWiM. There are 3 young people on the panel. 2 of them were quorum sensing Vibrio people. One of them from Bonnie Bassler's lab. Let's see. Christopher, I said Indiana University.
Casey.
Oh, Christopher says in TWiM 194, are the mini cells similar to apoptotic bodies or a bacterial exosome?
Ooh.
I would say, I don't know. It's a good question. Do bacteria do apoptosis?
I think they just die.
Casey,
and the others eat them.
Casey Adam from Sweden. Krista is a recent PhD working as an adjunct professor at a local community college. And she likes bacteria better than viruses.
Oh,
that's too bad. I'm sorry. Colin, let's see. Colin is working in probiotics. He works at a probiotic company. Recent story about Lactobacillus reuteri and autism. Loved it. Wow. Yeah, that's a very cool story. So he works on Myxococcus xanthus. It's a favorite bacteria of mine. And he sent 2 pictures of colonies attacking containment colonies that fell into a plate in one of my labs. Myxococcus, this is fellow out.
It's a slime mold. It's gliding motility.
Yeah,
and it eats other bacteria.
John Kirby, you know John Kirby?
Yes.
He works on it. He's over at Medical College of Wisconsin. Yeah.
It's not as pretty as Stigmatella. Stigmatella is another myxobacterium.
These are pretty pictures.
Yeah. It's a gorgeous microbe, and when they make fruiting bodies, it's quite spectacular.
And we had Jesse Madison. Rebecca is a PhD student in robotics at the University of Pennsylvania who's listening to TWiM. That's great. My favorite podcast. Camilla, we did her work on mini cells as garbage disposal. Camilla's at UCSD. Now, Camilla, since you were on that paper, why don't you answer the question from Christopher, who said, are the mini cells similar to apoptotic bodies or a bacterial exosome? I presume Camilla will listen because you want to know if you won, right?
Won the book. That's right.
Patrick is a grad student at Baylor in Waco. Yeah, there's 2 Baylors there in Texas. He recommends a book, Did You Just Eat That? A couple of researchers designed various microbiology experiments to test things like the 5-second rule and the effectiveness of double dipping in transmitting bacteria. I know listener picks are for TWiV, but I couldn't resist. No problem. You can always send in. Janet wrote in, Victor said, I'm not a huge fan of the term bugs, but a huge fan of free books. I agree. I don't like bugs.
No one does. But we have to play to the vernacular.
Yeah, that's what the name of the book is, and I didn't title it. I would not. Polly is entering from Los Angeles area. Ryan from Atlanta, 45 degrees Fahrenheit. Non-traditional career changer working towards medical school who listens to the podcast. Leandro Grant, a biochemist at Stockholm University. Erica has a bachelor's in English. Johannes from Perth. Erin Clare has sent in an email, loves to listen to the podcast. Ezra from Glasgow, UK. Angela, Trudy, Sophia from Greece recommends a book, How Microbes Rule the World by Bernard Dixon. Okay. Edgar loves the podcast. Sarah, who's in Christchurch, New Zealand. Sarah writes in to TWiP a lot. Brianne is in Victoria. That must be Canada, right? Yeah, BC.
Mm-hmm.
Rachel from Dublin. Lauren says, if I win the book, I'll let Elio borrow it since I live in San Diego. I just want it back before the summer.
Oh, God.
Here's some random questions. Is there a link between chronic ear infections and learning disabilities?
If porphyromonas is involved.
I don't know. I'm not aware of any, but if anyone knows, let us know.
Yeah.
Trinity. William is at the University of California, Santa Barbara. Nice place. Kevin is interested in phages and antibiotic resistance, treating bacteria. David is from Alexandria, Virginia, a post-bac student in computer science who wants to figure out how to use machine learning with computer vision and apply it to biology. All right. Mm-hmm. So we got 41 people writing in, and we usually don't get so many emails because people want the book. So I'll have to do this again in the future. So let me do a random number. I have my favorite random number generator between 1 and 41. And let's see, generate the number. It's number 33. I'll have to count down. I'm going to have to do that because I have to announce it. So we go 1, 2, 3, 4, 5, 15. Maybe I should just count from the bottom. How's that? That's smart. 41, 40, 39, 37, 36, 34, 33 is Edgar. So Edgar is the winner. He said, thank you for providing this podcast. It has sparked my interest regarding the microbial world and has helped me guide to pursue my major. Your collective dedication of providing the world with knowledge is truly awesome. Well, Edgar, you're going to get a lot of knowledge from this book. And to get it, you just need to send me an email, twim@microbe.tv with your address. If you live outside of the U.S., I'll need a phone number too. So Edgar is the winner. We'll have more books. We appreciate your participation. We will have more books to give away. All right, that's TWiM 195. Please, if you listen on a phone or a tablet, you use an app of some kind. Do you use a podcast app, Michael?
I do. I use the Apple Podcast app.
Apple Podcast, you can subscribe. You find a podcast you want, you just hit subscribe. That's good for us. It gives us hard numbers. It tells us how many people are listening. So if you can, please do that. And if you really like what we do, consider contributing financially. If you go to microbe.tv/contribute, there are a couple of ways you can do that, including, giving a $1 a month, either at Patreon or PayPal. It really helps us with our expenses. And of course, questions and comments, go to twim@microbe.tv. My co-host today has been Michael Schmidt from the Medical University of South Carolina. Thank you, Michael.
Thanks, Vincent. Thank you, everyone, for listening.
And take care of that P. gingivalis.
I tell you, I want to go to my dentist now for sure. I'm going to definitely floss, though.
Oh, they said in the paper when you floss, you get a bacteremia, and that could be how you introduce P. gingivalis into your blood, right?
Well, you do indeed get a bacteremia twice each day from brushing your teeth. You remain bacteremic for about 4 hours subsequent to brushing your teeth. So if you brush your teeth twice each day, you're bacteremic for 8 hours a day. But our immune system is quite efficient at trimming them out.
But that could be a way that P. gingivalis gets
that could be a way
into the brain, right? But still, flossing is good, right?
Flossing is good. You want your pocket depth to be toned like your 8-pack abs. 8-pack abs, that's what you got to have for your gum line.
That could be a title.
8-pack abs.
All right. I'm Vincent Racaniello. You can find me at virology.ws. Thanks to ASM for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.